Hi everyone, welcome to my channel Pure Biology, and today's topic is the transcription process in the prokaryotes. Before going to this topic, I want to say that the enzymes which are responsible for the transcription process and the terms that are related to the transcription process, I have already explained in my previous video. You just check out that one, and after that follow this one. So this is the transcription, and obviously, sorry for the disturbance. So how the transcription process is occurring in the prokaryotes, today we learn here. Okay. So first initiation process, here just like replication, the three processes are involved here: initiation, elongation and the termination. Okay. So first one, that is the initiation, and initiation starts when the phosphodiester bond is formed between the two nucleotides in an RNA. Okay. So, first the phosphodiester bond formation is occurring between the two nucleotides of an RNA. Second one, the RNA pol that binds at the promoter region. And which one is the promoter region? That is the actual place of the DNA, that is generally made up of the six base pairs, six bases are there. Okay, nitrogen bases are there. So this is the actual place where the RNA polymers can bind in the DNA strand. Okay, and here the main role, actually which one is recognized, the promoter region is how recognized. Then here is important role is found in the sigma factor. Okay, I totally already described in my previous video. You just check out that. So what happened? The sigma factor generally, before the functioning of the sigma factor, first you have to learn the RNA pol that bind with the promoter region to form the binary complex. Obviously the closed binary complex. Okay, closed binary complex. In the third part, what happened? The RNA factors, here, what is the role of the RNA factor? That changes the DNA binding properties of the RNA polymerase. Okay, it actually helps to change the DNA binding properties of the RNA polymerase so that its affinity increases to bind to the promoter region. That means its configuration level is totally changed by the sigma factor to identify or to recognize the promoter region of the DNA. That means it helps the RNA polymerase to bind the promoter region of the DNA strand to continue the transcription process. Next one, the transcription bubble is created by the local unwinding factor, obviously, and that begins at the site bound by the RNA pol. The area that is totally bound by the RNA pol, you just follow it. The area that is totally bound by the RNA pol is actual place which unwind after binding of the RNA pol, and after unwinding it formed a bubble-like structure, and this is the situation. You can say that this is the open binary complex. Okay, you can say this one the closed binary complex when the unwinding process is not done, and this is called the open binary complex where the unwinding process is completely done. Third one, this is the ternary complex. How the ternary complex is formed? The first two nucleotides incorporate and the phosphodiester bond form. Okay, and the ternary complex form here. So you can say this is ternary complex because here the RNA is seen and the DNA strand is seen and the RNA polymerase is seen. Okay, three complexes, three total substances are seen here, and that's why this is called the ternary complex. And here you can see obviously the RNA. You can see this is the RNA that forming. The next one, what happened? The holoenzyme that initiates the transcription process. You can know the holoenzyme when the RNA polymerase that containing the sigma factor, you can say this is the holoenzyme. Okay, and this initiates the transcription process. Okay, but the core enzyme, its function is that to synthesize the RNA on DNA template. Its function, Actually, holoenzyme is made up of the sigma factor and the core enzyme. And what is the role of the core enzyme? It helps to synthesize the RNA. And what is the role of the sigma factor? That it recognizes the specific promoter region, and which is totally suitable for binding the RNA ball on the DNA. Okay, so what is the role of the core enzyme? That helps to synthesize the RNA on the DNA template. Here you can see that synthesis of the RNA already occurring. In the next case, the sigma factor generally release the sigma factor, which one is attached with the core enzyme to form the holoenzyme. The sigma factor is released from this core enzyme in the next step, and when the base pair 8 to 9, okay, when the RNA chain that reaches the 8 to 9 bases, and what happened there? This core enzyme to undertake the elongation process. Sigma releases here, and the core enzyme that attached already with the DNA strand that continue the transcription process, and here the role of the sigma factor is not necessary. Okay, so holoenzyme that remains at the promoter. So holoenzyme only needed to bind the promoter region, but here the important function totally of the transcription process is totally done by the core enzyme. So the core enzyme that already continuously it binds with this strand, but sigma factor that already bind here that release in the next step. And initiation phase ends when the enzyme succeeds in extending the chain 10 pp. And before completion of the 10 base pair, you cannot say the transcription process is continuing or not, because before forming of the 10 bp, I explained it after that. Before continuing the 10 bp, the transcription process is not continuing. That means until the 10 bp chain is not elongated for this transcription process, you cannot say the transcription process is continuing. Okay, because it can stop or the transcription process is totally stopped. Okay, I will discuss this after that. So here, first in the transcripts that at the 5' end and is PPG or PPA, and there is another difference between the transcription and the DNA replication, that in case of DNA replication, primer is very important. Without primer, the replication process is not done. But here the holoenzyme is not necessary. In case of holoenzyme means not the sigma factor. This is not necessary for continuing the transcription process. But the core enzyme is important for continuing the transcription process. Okay, so this is the process of the initiation. So what you can understand here, first one, it remains in simple position on the template situation. First, what happened? The RNA pole that binds to the promoter region and to form the closed binary complex. In the next step, it unwinds and to form a bubble-like structure, and then you can say that open binary complex. And the third one, with the sigma with the holoenzyme, and obviously the unwinding bubble form template is present there, and obviously the transcription process starts. That means the mRNA chain you can see here. Then you can say that the already ternary complex is formed. And the next step, the sigma factor that already attached with the holoenzyme that release, and the core enzyme totally responsible or undertake the total responsibility of the transcription process or forming the mRNA chain. So this is the transcription process initiation, and the two parts that are important, that is elongation and the termination. Okay, we learn here after that. Okay, so this is the now elongation. Okay, before going to the elongation and the termination process, I just want to say that RNA synthesis is required, frequently aborted 2 or 3 bp, and this is called the abortive initiation. So I just said that if the RNA transcript that is continuing. We can see the process is continuing when it until that means it completes the 10 base pairs. If the 10 base pairs of the transcripts is completed, that means you can say that the transcription process will continue. Okay, and here obviously promoter clearance is important. If the promoter clearance is completely done, that means you sure that the transcription process will be continued. Okay, if this is not possible, that means the transcription totally stop, and this situation is called the abortive initiation. Okay. Next, we learn here the elongation process. What is the elongation process? Elongation process here, it involves the movement of the transcription bubble. The movement of the transcription bubble by a disruption of the DNA. Okay, the DNA templates totally disrupted and forming the mRNA. Okay, so what happened? The bubble moves from one place to another place, and the elongation process is continuing. Here the disruption of the DNA is first you see, and slowly you can see that the chain of the transcripts that completely elongated continuously or gradually. Here the enzyme moves, it unwinds the DNA to expose the new segment. The enzyme as usually moves towards this transcription process is done generally 5' to 3' direction. So as the enzyme moves from one place to gradually towards its end or 3' end, the new segment exposes. Okay, DNA unwinds slowly and the new segment exposes. Clear? Third one, the nucleotides that covalently added to the 3' end of the growing RNA chain. I am saying that 3' end of the growing RNA chain. And during each binding, the beta and the gamma phosphates are removed from the incoming nucleotides, that is from the DNA strand obviously, and hydroxyl group removed from the 3' carbon of present nucleotides at the end of the chain. That is, suppose this is the chain. Okay, this is the chain, and starting from here you just see that. So what happened? As the chain elongates slowly, its end part, that is its end part, suppose that is already present there. So what happened? How the bond is formed? The bond is formed, the beta and the gamma phosphates are removed from the incoming nucleotides. From the incoming nucleotides, the beta and the gamma phosphates are removed. And the hydroxyl group that is removed from where? The 3' carbon of the present nucleotide at the end of the chain. Suppose this is the end of the chain. Its hydroxyl group, here the nucleotide is present, and its hydroxyl group is removed, and its beta and the gamma phosphates that totally removes. Then this is attached with that. Here, and the chain elongates. And the RNA polymerase, when transcribes, it generates the positive supercoils and leaves the negative supercoils behind. What is saying here? When the transcription process is continuing, the DNA first unwinds. Okay, DNA first unwinds. So unwinding, in that situation, when the DNA unwinds, then the transcription process becomes starts. And if that area, suppose that area is totally involved in the formation of the transcription process. So when the transcripts is formed from that area, what happened? The RNA pol, when continuously transcribes towards the DNA chain, the positive supercoils is initiated or positive supercoils is formed ahead. Slowly positive, after completion of that area, then positive supercoil form and DNA become double helix form. Okay, and it leaves the negative supercoils behind, and negative supercoils. That means it continue to form the negative supercoils, and it's after completion of that area, then positive supercoils is formed. Suppose this is the area. So here the transcription process completion. So this area become joined again. Okay, this area become joined again. Okay, so it unwinds and then again supercoil is formed. So this is very important, and RNA polymerase is involved here. That it generates the positive supercoils ahead, obviously ahead, and leaves the negative supercoils behind. Clear? Leaves the negative supercoils behind. And here another two enzymes, these are very important, that is zinase and the topoisomerase 1. They obviously take part here or participate here. So this is the elongation process. What you just learned here, obviously elongation process. This process is very important here, where the coming nucleotides, its beta and gamma phosphate, that totally removed, and the hydroxyl group of the present nucleotide, that is removed, and then the bond formed between the coming one and the present one. Then the elongation is completed. Okay. After continuing this elongation process, what happened? Some sequence that is present on the DNA, this is very important, that say that transcription must be stopped here. These are called the terminators. These This sequence is very important to stop the transcription process. And what happened here? The dissociation of the polymerase is seen by the stabilizing the attachment of the growing transcripts. What happened here? The dissociation of the polymerase. Polymerase is totally dissociated from the chain, DNA chain. Okay, and the transcripts form hairpin-like structure. Okay, transcripts, mRNA transcripts that form the hairpin-like structure, and its loop that containing GC rich base pairs and followed by the U rich base pairs. Okay, so this structure is the hairpin-like structure. So here the rho factor is not important. Okay, some sequence that present on the DNA that generally try to stop the transcription process, and this is known as the terminator. Sometimes this terminator is not important. Sometimes the rho factor, that is very important, and its generally activity is dependent on the ATP. Okay, what is the actual role of this rho factor? This rho factor actually disrupts the DNA RNA complex. This complex is totally disrupted by the presence of this factor. Okay, and the RNA that released from this bonding. Okay, RNA is released from this complex. So this is the termination process, and this is the elongation process. You completely I explained here properly with the proper diagram and rough, obviously not proper. This is the rough diagram. I am trying to explain is properly, and I think you can understand it properly, and obviously you consult any book that is related to the genetics and after seeing my video, so it will be helpful to you. Okay, so this is the transcription process of the prokaryotes, and I will try to give the transcription process of related eukaryotes in my next video. So please continue with me, and the new ones obviously try to subscribe my channel for the growth of this one. Okay, and obviously again I thankful to those who already have subscribed my channel. Okay, thank you.